Our final speaker today is Scott Lowe. Most of you probably know Scott as my postdoctoral advisor. It turns out, though, he had this entire career before we met and has tried to carry on after I left, so deserves some credit for that. So Scott was an undergraduate at the University of Wisconsin. He then came to do his biology graduate work at MIT, where he worked with Earl Ruley, and he did brief postdoctoral work with Tyler Jacks and David Hausman. He then, like Phil Sharp, moved to Cold Spring Harbor to work on DNA tumor viruses, particularly adenoviral UNA. More recently, Scott's moved to New York City to join the Memorial Sloan-Kettering Cancer Center, where he is currently the Associate Director for Basic Cancer Research, as well as the Chair of the Geoffrey Beene Cancer Research Center and an HHMI investigator. So welcome, Scott.
All right, well, while we're getting set up, I just want to thank the organizers, Phil, Sangeeta, and Tyler for hosting this event. It's always good to be back here at MIT with so many friends, colleagues, and former trainees, even though some of them are ne'er-do-wells. Anyway, I guess I was invited to this symposium certainly not an RNA biology expert, but I'm a user of RNA biology, and we've spent a lot of time in my lab trying to harness RNAi to characterize cancer genes, and that's what I'll talk about today. I think probably what I'll show you is a paradigm of the questions that we're interested in my laboratory, but I think are really reflective of the type of questions cancer biologists and geneticists are interested in general. We're interested in trying to understand the genes and processes by which normal cells become cancer cells, the so-called drivers whose alteration leads to the malignant state. And we're interested in this for two reasons. On one hand, cancer drivers alter key cellular processes, proliferation, survival, and so on, and affect key nodes in these processes, so we learn a lot about basic biology. But of course, we're also interested in trying to understand these drivers because we hope that by understanding the molecular origins of cancer, we can ultimately treat it. So many of us are also interested in what I like to call tumor maintenance genes or cancer dependencies. These would be genes that are required to maintain the malignant state and whose inhibition could lead to essentially the differentiation of a cancer cell or its death or senescence. Now, I'm really inspired by my time here at MIT. I got interested in and appreciated the fact that cancer doesn't occur in isolation. It occurs in the context of a tissue microenvironment. And as such, the processes that go on within the microenvironment can have a dramatic impact on this paradigm. And so we, for now some years, have been interested in trying to understand cancer in vivo in the context of genetically engineered mouse models. Now, we've also taken advantage of RNA interference, and again, this, I think, is really relevant to the topic today, to interrogate these processes. And if one thinks about it in this paradigm, one can imagine, in this case, I'm talking primarily about short hairpin RNAs that can stably suppress gene function, as a way to characterize some types of drivers, tumor suppressor genes, these genes whose suppression can promote tumor development. And one can also use RNAi to try to understand cancer maintenance genes, genes whose inhibition in an established tumor will lead to its elimination or its loss of malignancy. And again, we in many, many other laboratories have exploited RNAi for these purposes. Now, I want to introduce the platform of RNAi that I'll talk about through the rest of my presentation. And this is really introduced to me and pioneered in large part by Greg Hanna, my former colleague at Cold Spring Harbor, together with Steve Elledge at Harvard Medical School. And our adaptation of it sort of involves three components that I'll go into here. We express our short hairpin RNAs often from an inducible promoter, very often the TRE promoter, allowing sort of tetracycline or doxycycline regulation of the shRNA. Our shRNAs are linked to a fluorescent reporter that allows us to track expression within cells and ultimately, as you'll see, within tissues. And we use a microRNA-based platform to trigger the RNAi machinery. Ours was initially based on the Mir30 design, which we believe naturally enters the RNAi machinery and may be less prone to sort of promoting off-target effects. More recently, work from a former postdoc and former student in mine have developed an adaptation which actually reflects some biology also discovered in Dave Bartel's lab that leads to more efficient processing of these shRNAs and better knockdown. Now, we've used RNA a lot, and there's been a lot of discussion about the utility of RNAi and perhaps problems associated with its use and off-target effects, again, confounding the interpretation of studies. And so we've spent a fair amount of time trying to optimize this platform of RNAi to try to get triggering of the RNAi machinery at relatively low dose and high specificity in order to minimize theoretically and often practically off-target effects. One of the assays that we developed, we call the sensor assay. I don't have time to go into the details, but it's a high-throughput way in which we can use biology to sort out for shRNAs that are highly potent using a reporter assay, very much like the 1x1 one uses to characterize microRNA targeting, for example, 3' UTRs. And using this, we can sort out good from bad shRNAs. And when we say good, we're interested in those that are working really when we express them at only 1 copy per cell. And this will be important later on in my talk. And so here's an example where we've tried to target p53, where the score, if it's good, is indicated here. If it's bad, it's a negative score. And if we do a Western blot, you can see the good ones get very good knockdown of p53, 1 copy per cell, the bad ones less so. I should point out, if we crank up the expression by, for example, high multiplicity of infection transduction of these, even the bad ones will knock down, but of course we worry they may do other things as well. Now, we did an experiment where we took these shRNAs targeting p53, either the potent ones or the weak ones, and we introduced them into p53 null cells. And then we did transcriptional profiling of the cells following expression of the shRNA. Again, there should be no on-target changes in gene expression because p53 is not expressed. So anything we see should be due to an off-target effect. And so again, this I'm not sure how well it projects, but we have single copy, 1 copy per cell, or high copy, and this is no shRNA. And what you'll see quite clearly is as we increase the dose of these shRNAs, whether or not they're potent or not, we start to see more off-target effects. And again, at low copy number, for most of these, again, not apparent at this resolution, we only see a couple, if any, significantly differentially expressed genes. So this makes sense. We've also looked at small RNA species. Again, one thing one worries about with RNAi is it might clog up microRNA processing and therefore lead to some sort of toxicity. And so here we've looked at small RNAs, again, where we pooled our potent and weak shRNAs targeting p53 at single copy and high copy. And really what we see is the only ones that are sort of differentially expressed when we go to high copy are actually the ones we programmed within the cells. There isn't much, again, differences in microRNA biogenesis, even at high copy number. And if we compare the single copy shRNAs, again, that have potent effects on the cell, compared to no shRNA, you can see very little, if any, change in the abundance of endogenous microRNAs, again, suggesting that these shRNAs are not perturbing the RNAi machinery. So we've used this platform, again, to do lots of things. Screens, as many other labs have, to identify new genes that might modulate cancer phenotypes. But I'm really going to spend the rest of my time talking about a different approach, where we've tried to use RNAi to study the function of genes in vivo. And in principle, one can imagine with an inducible RNAi system, one could turn on and off a gene at any time, at any place, and for any length of time in vivo, and that's been our goal. So here's the system. This is actually built initially on concepts developed in the Yanish lab, again, just across the street, using ES cells where we could very efficiently target the same genomic locus. And what we've done is to use these to target in an shRNA vector, again, the TRE promoter driving an shRNA of interest linked to GFP. And in these ES cells, there is an RTTA that enables regulation of this shRNA that's either tissue-specific or ubiquitous. Now, this allows very rapid production of mice that will have doxycycline-inducible shRNA expression. One can induce it in the embryo if one wants to study the role of a gene during embryonic development, but what if one does not? One can wait till the mice are born and then induce the shRNA knockdown in the adult animal. One can also cross these strains into other cancer-prone strains and look at how perturbing gene function might affect the trajectory of a particular cancer, or for that matter, any disease, if there's a particular disease gene. Now, this is a powerful system because, again, it's very easy to study any gene. We're not making a new targeting cassette. We're just changing the shRNA. It's inducible. And again, we can do either widespread, ubiquitous, or tissue-specific gene expression, depending on the nature of the RTTRA and where we induce shRNA expression. So I'm going to show you a couple examples of how we've used this technology. I should say that all of this technology can easily be adapted to the CRISPR-Cas system that we've used before, and we've done this. I won't talk about that today. But one thing that's different about RNAi is that it does not disrupt the endogenous locus, and so as a consequence, it's reversible. And I think you'll see the power of that during the course of my talk. So one thing we've tried to use this technology, you think about it, if we can make a mouse very easily and knock down a gene in vivo, we could imagine what might be the consequences, the phenotype of inhibiting that gene, and if it were a drug target, would that lead to any sort of toxicity? And so a molecule we've been particularly interested in is a protein called BRD4. This is a member of the bromo-X-terminal family. It is a histone reader. It binds acetyl-lysine. And we got excited about this because we identified it as a gene that might be a therapeutic target in myeloid leukemia. A number of other groups here in town identified it as a therapeutic target as well. It was particularly interesting because Jay Bradner and others had developed small molecule inhibitors of this, and so we could immediately ask whether or not those small molecules might have activity in leukemia, and they did. But one of the initial problems, and these drugs are in clinical trials and they're getting much better, is that the initial drugs had a relatively short half-life. And so while we could see there were anti-cancer effects, it was very difficult to assess what might be the potential toxicities of sustained inactivation of these BET proteins because the drug really wasn't around long enough to do it. So we made one of these shRNA transgenic mice. This is the configuration shown here, different BRD4 shRNAs. And here's just an example of the type of knockdown we get. These are actually cells from the intestine. Ren stands for shRNA targeting renylluciferase. It's a negative control. It doesn't knock down any gene. We use it in all of our experiments. If we add doxycycline, again, if we have two different hairpins targeting BRD4, we get good knockdown. GFP comes up as we predict. And if we take away doxycycline, it's reversible. Go back to endogenous levels of the protein. If we ubiquitously knock down BRD4 in the adult animal and wait 2 weeks, this is actually interestingly, as I've been told, about the length of a typical toxicity assay sort of as in drug development, we don't see really much going on. But it doesn't stay that way. You can see quite clearly by 7 weeks now there's a pretty dramatic phenotype, quite pronounced loss of hair and sort of a ruffling of the skin. Now, it's a very interesting phenotype. We haven't characterized it in great detail, but one thing I'll just point out is we see an epidermal hyperplasia. And that seems to obstruct the hair follicle, and presumably that's why the mice lose the hair, although there could be other explanations as well. Nevertheless, this isn't a phenotype one would immediately imagine would happen when you inhibited a gene or a protein that would be essential for cancer. Again, here in this case, we're actually getting a hyperplasia. We've also seen phenotypes in the intestine, and because I'll talk about the intestine later in my presentation, I just want to give you a little background here. This is a schematic diagram of the mouse small intestine. Many of you may be familiar with it. This is a highly regenerative tissue. Normally, cells proliferate. The stem cells are in the crypt. There's proliferation, transient amplifying cells. You can see the proliferation here. The cells differentiate and eventually die and slough off into the lumen. You can measure the differentiation using alkaline phosphatase, or for example, here's a marker, keratin 20. So again, a very structured organ. When we looked at the mice where BRD4 was expressed, BRD4 shRNAs were expressed, we noticed, and you may not even be able to see it, sort of just something different about the morphology of these small intestines. which became clear when we started to do immunohistochemistry or in situ hybridization for markers of different cell types. So lysozyme measures the Panna cells normally in the crypt. OLFM4 measures stem cells, and Alcian blue measures goblet cells, again, interspersed throughout the villi. But you can see when we knock down BRD4, there's sort of a depletion or disorganization of the cells in the intestine, something that is potentially abnormal. Now, most of the mice are fine, but some of the mice, in addition to losing hair, lose weight. We assume that it's in part, or it's probably due to this intestinal phenotype. Here's an example of a mouse here where we've added dox. They grow for a while, and then they start to lose weight. Here it is hairless. But now we can do an experiment you can't do with standard genetics, and you can't do with CRISPRs either as well, which is to take these mice off doxycycline and ask what happens. So if we do this, we see immediately that the mice begin to gain weight, and within the matter of a couple of weeks, all of the hair is regained. If we look in the intestine, again, it's hard to see here, the morphology returns to normal, and if one looks at the different cell types, here we have BRD4 suppressed, here 2 weeks after it's back on, everything goes back to normal. And so we notice that we interpret these results to say that inhibition of BRD4 24/7 could produce some toxicities, but they're reversible. We did get concerned about how these effects might play out if mice were treated with DNA-damaging agents, which are known to affect the intestine as well. And so we did an experiment where we added dox to suppress BRD4, waited 2 weeks, and then gave a sublethal dose of irradiation. What we noticed, as would expect, is the control mice started to lose weight, but then that plateaued, whereas those where we suppressed BRD4 continued to lose weight. There were dramatic differences if we look histologically in the intestine. Normally, there's initial proliferation that ultimately resolves, but you can see the two mice, the two shRNAs where we knock down BRD4, there's a problem. That's really quite apparent when one looks at Ki67 to look at proliferation. Again, this initial burst of proliferation during organ regeneration is disrupted when BRD4 is suppressed. And as a consequence, these mice die. So the dose of radiation that normal mice tolerate, when we suppress BRD4, they're very sick. And so we can learn a lot from this sort of system about sort of the potential things that oncologists should be thinking about as they begin to treat patients with BET inhibitors. Of course, we may be underestimating the toxicities because BET inhibitors hit more than one family member, where here we're knocking down BRD4. But we learned that, probably not surprisingly, but something we always have to keep in mind, that these chromatin modulators have very diverse effects, sort of almost opposite effects depending on the tissue and the context. In this case, the effects seem to be reversible, but we do worry that there might be synergistic toxicities with DNA damage and BRD4 inhibition. Okay, I'm going to spend the last 10 minutes of my talk talking about another way in which we can use these mouse models to interrogate cancer gene function or cancer relevant processes. And here what we've done is to spend a lot of time in targeting tumor suppressor genes. And the concept here is we know that inactivation of a tumor suppressor can contribute to cancer from work in genetically engineered mice. But it's very often hard to know whether or not that cancer requires ongoing inactivation of the tumor suppressor and ongoing deregulation of the pathway to sustain the malignant phenotype. And so we've targeted a number of tumor suppressors. I'm just going to talk about one, which is APC. Now, this gene is probably familiar to all of you. It is a negative regulator. It encodes a negative regulator of the Wnt pathway. Germline mutations in APC give rise to familial adenomatosis polyposis, or polyps in the colon. And human colorectal cancer, about 80% of those have inactivating mutations in the APC gene. Just to put it in perspective, mutations in APC, given the abundance of and the magnitude of mortality from colorectal cancer, actually give rise to more cancer deaths than activating mutations in KRAS in the lung and the pancreas combined. So again, this is an important pathway. Its deregulation is associated with a lot of cancer morbidity. When inactivated, again, I go back here. It normally acts to stable as in a complex with beta-catenin, and when mutated, beta-catenin is released to go into the nucleus where it can drive expression of Wnt target genes, for example, MYC and AXIN2. The mice that are knocked out for APC develop polyps. They develop them in the small intestine, which doesn't quite recapitulate human colorectal cancer, but nonetheless, they do get disease. And the knockout mouse, constitutive knockout mouse, is embryonic lethal. So we made mice where we could acutely suppress APC expression throughout the intestine. I should say, we really have to have good shRNAs. We learned this through some trial and error. If you don't get rid of about 98 to 100% of APC, there's very little phenotype. But once the shRNAs are very good, we see very striking phenotypes, some shown here. This is our control shRNA. Here we've added doxycycline to knock down APC. And within 10 days, you can see this massive proliferation throughout the small intestine. And we see proliferative cells up in all the villi where they're not normally supposed to be. This comes at the expense of differentiated cells and enterocytes. You see less of those, and sort of an expansion of a less differentiated or non-differentiated cell. If one looks at stem cell markers following acute APC suppression, we can see again stem cells or cells with properties of stem cells start to be located in aberrant positions. Here we're looking at OFLM or LGR5, another marker of stem cells. And the Panna cells, which are stained with lysozyme, are also now in aberrant places. Now, importantly, the effects of APC suppression on this phenotype are completely reversible. So if we remove doxycycline after 10 days, everything goes back to normal. And you can see that just looking at proliferation here. And we can look at target gene expression, again, isolating intestinal epithelium. Here we're looking at Wnt targets. Add dox, these genes become deregulated. Take it away, it goes back to normal. And again, if we look at markers of stem cells, again, a very similar pattern. This is just a Western blot to show you, as I sort of alluded, we get very good knockdown of APC. Again, here's day 0. 2 days, basically it's gone, inversely associated with GFP. So this is great. We can see that suppression of APC causes a phenotype. We knew this would happen from the null, but that it's reversible. But we really wanted to study cancer, and these mice died of weight loss and intestinal dysfunction before they could develop any disease. So we had to develop a system where we could activate our shRNA in a mosaic way in only a subset of the intestinal epithelial cells. The approach that we took involved 3 alleles, one of them generated in our laboratory. And the way it works is like this. We have our tetracycline responsive shRNA, expresses APC or control. We have a RTTA needed to regulate that, but it's kept silenced by a lock-stop LOX cassette. We also have intestinal stem cell promoter LGR5 driving a Cre system, a Cre-ER. This can be activated by tamoxifen, and the idea is, initially shown by Hans Clavers, if one adds a limited amount of tamoxifen, only a subset of the cells recombine the RTTA. They're now on a strong ubiquitous promoter, where in the presence of doxycycline, we can induce the shRNA. So the idea is, if we activate this in the LGR5 positive stem cells, these cells will be lineage traced and capable of inducing our APC shRNA. This is the model, what it should look like. This is when we do IHC for GFP. Here's sort of a different view of this. This is the small intestine. This is the large intestine. Again, you can see these foci of GFP positive crypts. Now, one thing we were quite excited about when we noticed this is that not only did we see spots in the small intestine, but we also saw them in the large intestine. Remember, I told you the genetically engineered mice, for reasons we don't understand, only get polyps in the small intestine, but people get tumors in the large intestine. Now, these can be stable. This is just in black and white, where we've kept mice on doxycycline just with the control shRNA for 40 weeks. So we really are activating the system in cells that are the stem cells. Now, the advantage of being able to turn the system on in the large intestine is we can do mouse colonoscopy. My postdoc loves to get up on Monday morning and do this every week. But this is what it looks like. Here, what we've done is we've knocked down APC sporadically throughout the large intestine for 8 weeks. And you can see, as we would predict, these mice get tumors, polyps. Because the shRNA is linked to GFP, you can see they're also green. So we can track longitudinally in these animals essentially polyp formation, their expansion, and in principle, their regression. We did some gene expression profiling. I won't dwell on it, but one thing we noticed right away is if we look at sort of its similarity to other gene expression profiles, these colon polyps where we've knocked down APC have a very highly significant correlation to what has been described in human colorectal cancer. So again, we think we're getting the right disease. Now, of course, the exciting thing here is we can ask in these established disease, if we re-establish APC expression, what happens. And so here is an example of two different mice where you can see that these malignancies regress. It takes a varying amount of time. In some cases, it can be very rapid. In some cases, it appears incomplete. But when we sacrifice these animals and look at these sort of scars, we really only see normal epithelium. So the lesion is involuted but hasn't quite restored the normal tissue architecture. So what happens when we turn on APC? Well, the first thing we see in a matter of a day or two is that proliferation in these lesions halts. We can measure that again here by looking at DNA synthesis, BRDU incorporation. Using markers of differentiation, I'll only show you a few examples, we see massive differentiation shortly after reestablishment of APC and a return to tissue homeostasis. So here is a polyp, again, no differentiated enterocytes, few goblet cells, but again, throughout these adenomas, very rapidly we see effects. Now, the gene expression profile, in addition to give signatures of colorectal carcinoma, also showed signatures of immune cells and stromal cells and so on. So there's a lot going on when these tumors grow and then ultimately involute. And we wondered whether or not this requirement for APC suppression to sustain the malignancy was cell autonomous. And so to look at this, we took advantage of an organoid system developed by Hans Clavers. Again, many of you may be familiar with this technique, where one can isolate cells from the intestinal epithelium, put them the right growth factors, including R-spawn and a Wnt ligand, and then form these organoid structures, which actually have a structure that mirrors what is seen in the intestine with a crypt, transient amplifying cells, differentiated cells, and then sloughing off into the lumen. So we looked at what would happen in isolated epithelium where we added doxycycline to knock down APC. This is in the absence of doxycycline. You see the normal organoid structure. And again, these spheroids form very dramatically and very rapidly when we knock down APC. A total lack of organization recapitulated in vivo. And here we're just looking at staining with EDU to mark DNA synthesis. And again, you see it throughout these organoids. But just as we saw in vivo, when we reestablish APC, the system goes back to normal. So again, here you're seeing proliferation largely in these crypt structures as in the controls. Now these are from the small intestine. We've also looked at those organoids derived from the colon. Again, when we knock down APC, you see this spheroid formation where basically all the cells are in cycle. And again, K20, keratin 20, is a differentiation marker. You don't see it because these cells are undifferentiated. But 4 days after reestablishing APC, again, you see this very beautiful organoid structure, again, proliferation in the crypts, these differentiated cells outside. So again, the effect of APC on sustaining this sort of aberrant proliferation and differentiation block is cell autonomous. Now, this was all very exciting, but of course, people, because it says that APC inactivation is needed to sustain polyps. And of course, one that would suggest that targeting this pathway would eliminate polyps. But of course, the surgeon can do that clinically. It's easy to cut out a polyp from a patient. That's not what kills patients with colorectal cancer. Instead, they die from advanced carcinoma. Now, that disease involves additional mutations. Work initially from Berg-Vogelstein and many others shows that activation of KRAS and inactivation of SMAD4P53 are important in progression of the disease. And I just have a couple more slides. And so it'd be interesting to know whether or not with these additional genetic changes, would we see the same result, or would these cells become independent of deregulated Wnt signaling? And this wasn't essentially just sort of a hypothesis. In other models, that has been shown to be the case, that, for example, alterations in RAS signaling can lead to a loss of dependence on aberrant Wnt signaling. So we did lots of crosses to bring in these other alleles, and here's just one where we're looking at colonoscopy with the APC suppression and KRAS activation. And you can see within a matter of a couple of weeks, we get these very massive polyps that by 6 weeks basically has occluded the colon so we can no longer do endoscopy. If one looks at the onset of disease, the green line is when we knock down APC alone. When we knock down APC in the presence of KRAS, that's the pink. And if we also incorporate mutant p53 alleles, null or mutation, we don't see any further acceleration. But the key question is, in these settings, where it's true malignancy, and I should point out, I don't have time to show it, if we transplant these cells into recipient mice, they're highly malignant metastatic colorectal carcinoma. So this is the real deal. And when we turn on APC in the system, you can see in a matter of a few weeks, these dramatic regressions. And again, we haven't taken the colonoscopy out forever, but we've never, ever seen a relapse of the disease in these mice, out to now many, many weeks. Again, we see that establishment of APC and reestablishment of control of Wnt signaling forces differentiation of these cells and returns the tissue to sort of homeostasis. So it isn't that just all the cells die, they go back to normal. And that's associated, again, with suppression of Wnt signaling and the stem cell signature that we see in this system. This is my last slide. I just want to show you, we have also seen locally invasive disease in our mice when there's KRAS or KRAS and mutant p53. And here's an example where we've reestablished APC in a mouse that had these 3 alleles. And we see basically normal intestinal epithelial cells outside their normal location, suggesting that they invaded and then differentiated and came back to be normal. We don't know whether they're sustained, they lack any proliferation. So again, this suggests that APC restoration can also block the progression of metastatic disease. So then just to finish, and I'm sorry I went a few minutes over time, basically we believe that APC inactivation and hence deregulated Wnt signaling can sustain malignancy by suppressing differentiation, and that's through a cell autonomous mechanism. And that despite the fact other lesions can cooperate with APC to promote advanced cancer, they don't alleviate, at least in these models, the dependence on pathway deregulation. Just to summarize the main people, again, Christoph Fellman has done a lot of the work on RNAi. Luke Dow is the main person that did the APC mouse. Jess, Nil, and Luke collaborated on the BRD4. We have collaborators, and so I'll stop there and thank you for sticking around and your attention.
Excellent. Thank you, Scott. That's a great way to end the power of RNAi to inform us about cancer biology and new approaches for therapy. It's my job to sort of bring the curtain down on the 2014 Koch Institute Symposium. As I was thinking about what I would say, I remembered that actually 10 years ago we had another symposium much along the same lines. We called it the revolution, the small RNA revolution. And at that point we sort of reflected where this field was going to go. And I think we were fairly accurate in knowing that microRNAs were going to continue to inform biology and disease, and we heard examples of that today, that RNAi was going to help us understand cancer pathways and how to treat the disease, and indeed, that both microRNAs and RNAi would help us ultimately treat directly. And we heard beautiful examples of that, including clinical examples of where diseases are being treated, not just in the liver, but now also in other sites. And this reflects, again, the combination of biology and increased understanding of these systems with engineering and, importantly, commercialization. And this is incredibly exciting, and I think this is just the tip of the iceberg. Clearly, the non-coding RNA world is much more complex, and we heard beautiful examples of long non-coding RNAs, and I think they, too, will have important therapeutic implications. And finally, the Cas9 system that Jennifer talked to us about is really changing everything remarkably rapidly. So that's the next revolution that's happening right before our eyes. It'll change biology for sure, and I'm sure as well it'll change medicine in the hands of biologists and importantly in the hands of engineers as well. So exciting times. Thank you to all the speakers for bringing those to life for us today. Back to the analogy about bringing the curtain down, these are productions, these Koch Institute symposia, and a good production requires many things. There are 4 things that I thought of. Firstly, you need good directors, and we had good directors in the case of Sangeeta and Phil. You need good cast members, and we had those in the case of our terrific speakers today. And you need a great backstage crew, and we had those as well. Pam DeFreya and Laurie Spindler, Cindy Quentz, Anna DeConnick, Martha Murcia, and so many others who contributed to today's activities. So if you don't mind, one more time, giving them all an applause for their efforts. So I said there were 4 things that make a good production. Those were 3 of them. The 4th is you need a good and attentive and appreciative audience, and that was you all. So I wanted to say thank you to all of you for coming and staying to the end. Remember earlier there was this sort of read and response participatory aspect of my comments in the morning, so let's do that again. Thanks, everybody. Well, I was looking for you're welcome, but that's okay. So we'll be back here again in about a year's time, talking about a subject to be determined. So stay tuned. Take care.